Hi, my name is Ashley Kennedy, and today I'll be talking about RTK signaling in cancers. First off, what is RTK? RTK stands for receptor tyrosine kinase. It is a cell membrane receptor with three domains. It has an intracellular domain called a tyrosine kinase domain, an intermembrane domain, and an extracellular domain where a ligand will bind. This particular receptor is responsible for the beginning of a signaling cascade. And due to that, it is highly regulated because the signal that it is sending will have some very large consequences for the cell because it will deal with regulation of cell growth and proliferation, cell differentiation, and cell survival. And what's important about this particular receptor is that it allows for extracellular to intracellular signaling. And there are a lot of different types of RTKs, and they all have receptor-specific ligands. Now, in normal RTK activation, as you can see here in this figure, a receptor-specific ligand is going to bind to the RTK. That in turn is going to cause two adjacent RTKs to dimerize, which will lead to trans-autophosphorylation of their tyrosine kinase domains. This phosphorylation of the tyrosine kinase domains will cause the activation of the signaling pathway and the recruitment of a variety of other proteins, which will then be phosphorylated to the tyrosine kinase domain. The result of the signaling activation is that transcription factors will be activated, which will lead to gene expression, and then that will lead to alterations within the cell metabolism. And this entire process is a normal cell function. However, it doesn't always happen the way that it's supposed to. And when it doesn't happen the way it's supposed to, that is aberrant or abnormal RTK activation. Now, there are two main types of aberrant activation. The first is gain of function. The second is amplification. As shown by this figure, gain of function means that the RTK receptor is being turned on and beginning the signaling cascade without a ligand binding. This could be caused by an alteration anywhere in the RTK receptor, but its end result is signaling activation without ligand binding. For amplification, in this particular figure, it shows that there are way more RTKs being expressed on the cell membrane than there should be, and therefore there's more signaling than what should normally happen. But the big point of amplification is that not only is the RTK signal being expressed, but it is being overexpressed in some way. Often, this abnormal activation will lead to cell death, but it is also very commonly seen in a lot of different cancers. So today, we're going to talk about aberrant RTK signaling in activation in cancers. Now, there are three very well-known signaling activation pathways that I'm going to go over, which is chromosomal rearrangements, kinase domain duplication, and autocrine activation. Lastly, we're going to go over a new and not very well-known type of novel activation via microRNA. The first aberrant activation pathway we're going to talk about is kinase domain duplication, which is a gain of function type of activation. It is when a single RTK is made with two tyrosine kinase domains. As you can see in this figure, kinase domain duplication literally means that there are two tyrosine kinase domains on a single receptor instead of one. Because there are two, this receptor is able to dimerize the tyrosine kinase domains, which will allow them to transphosphorylate and become activated. This type of activation does not need a ligand. It can happen spontaneously within the cell, and the result could be cell death. It could be cancer, or it could just be unregulated and continuous signaling activation in some way. This particular type of domain duplication has been implicated in a lot of different types of cancers. The next one we're going to talk about is autocrine activation, which is a type of amplification activation. In autocrine activation, you are basically having normal activation of the pathway, but you're having abnormal levels of activation. For example, in this figure, you can see that there are way more ligands than there usually would be. That means that the RTK receptor is being activated, and then when it goes through its normal pathway and it activates the cell signaling that it needs to activate and it goes to be turned off, it's just going to be reactivated. Like there's no way for the cell to turn off that outside signal. So the signaling cascade is going to be activated way more than it should be. Now, this particular type of activation has been seen in a lot of cancers, and it's linked to tumorigenesis. As for our third well-known pathway, we'll be talking about chromosomal rearrangements. And this is an important pathway because it can cause both types of activation. It can cause both a gain of function and it can cause amplification. Now, chromosomal rearrangements is a normal part of cell division. However, if there is a double strand break within the chromosome, that double strand break could happen within the gene coding for the RTK receptor. And when that gene is broken, a section of it might get deleted out. For example, in this figure, they're showing that the transmembrane domain of that gene was removed. Since the transmembrane domain was removed, it can no longer bind to the membrane. And since it can no longer bind to the membrane, when they start making these receptors, all they're making is the tyrosine kinase domain, and then they're basically allowing it to be released into the cytoplasm. And when it gets released into the cytoplasm, it is able to bind to another non-functional version of this particular RTK domain, and it's causing activation. And this is abnormal activation that is coming from within the cell, which means that is a type of gain of function because it's not requiring a ligand to turn it on. But if enough of these are made, enough of these abnormal ligands are made, or for example, if during chromosome rearrangement, the gene for the RTK is doubled, then you could have multiple copies of normal RTK, as shown on the left, being expressed on the membrane. That would be a form of amplification. And so chromosomal rearrangements can lead to multiple types of aberrant activation. It just depends upon whether the gene itself is being duplicated or whether it's being broken in some way. But the result is unregulated and continuous signaling activation in a lot of cases, abnormal levels of signaling activation, and this is another type of aberrant activation that is linked to cancer. And lastly, today we're going to talk about a novel type of activation which has to do with microRNA within the cell. This type of activation is a gain of function activation because the receptor is being turned on without a ligand binding. MicroRNAs can directly modulate RTK expression without the presence of the ligand. And this is important because microRNAs are already within the cell, so they can regulate RTK expression normally within the cell. And when they do their job properly and do normal amounts of regulation, then you will have normal activation of the cell in non-cancer cells. However, if this microRNA turns on the receptor and there's nothing to turn it off, or the microRNA is not degraded properly, then you will have lack of regulation of the RTK signaling pathway, and the signaling pathway will be turned on, and it will lead to abnormal signaling activation. This is seen most recently in cancer cells, and it is linked to tumorigenesis in recent research works. And that is all I have for you today. Thank you for listening, and let me know if you have any questions.